So today, we are going to take our concept of Mendelian genetics and expand it because, as with life, um, biology is not always as simple as we first understand it to be. Um, so odds are we won't finish this today. There's a lot of different things to talk about. Um, So we are just going to kind of take a leisurely stroll through this information, get as far as we can without getting too rushed. Just kind of letting this stuff soak in. Finishing up tomorrow, and I'll have some practice problems with this stuff tomorrow as well. Nothing too crazy. We are looking at. Let me check my notebook. Looking at practice tomorrow. We'll talk about human traits on Friday. We don't have school on Monday. We'll talk about chromosomal inheritance and like sex-linked stuff next Tuesday, Wednesday, Thursday. Uh, Practicing next Friday. Um, and then some more chromosomal stuff looking at progress questions Friday the 29th. Um, just so you have a generalized timeline. So we're going to stick with genetics for a hot minute um, and take our time. So, we are talking all about complex inheritance. We'll have a lot of fun examples. Um, Nothing too crazy. While it does say complex, it just means not Mendelian, essentially. So there's lots of different ways complex can be protrude. So what we've been talking about recently is degrees of dominance, right? So with Mendelian genetics, another way we could say Mendelian genetics is complete dominance, meaning if you have one dominant allele, you're gonna show the dominant phenotype. Period. End of story. Um, so complete dominance is where the phenotype of the heterozygote and the phenotype of the dominant homozygote are indistinguishable. So if you're writing this down, you really only need the definition of complete dominance. So when we talk about Mendelian genetics, we're talking about complete dominance. But there are other sort of degrees of dominance, like everything in life. Genetics isn't black or white, most of the time, anyway. Complete dominance is not something that happens in humans a lot. Like I said, we've got stuff like widow's peaks being complete dominance. Polydactyly, I believe, having an extra finger or toe is complete dominance. Attached or unattached earlobes is complete dominance. But I believe that's pretty much the gambit. Everything else has some sort of not external, but some sort of different system that makes it work. I will most likely use me and my family as an example for a lot of this stuff. I have a little brother and a little sister, so just in case that pops up, we look nothing alike. So that's degrees of dominance. The two big ones when it comes to complex inheritance that y'all have probably heard of are incomplete and codominance. So incomplete dominance is when the heterozygote has a phenotype that is somewhere in between our dominant and recessive phenotypes. So when a red snapdragon is crossed with a white snapdragon, all of the heterozygotes are pink. I believe there are multiple genes that interact with this, but hair texture is incomplete dominance. So, like if one parent has really, really curly hair and one parent has thick, straight hair, odds are their offspring are going to be somewhere in between. I think dog tail length is also incomplete dominance. And bunny fur texture is also incomplete dominance. So if we had a bunny with really, really long hair and a bunny with really, really short hair, and we cross them together, we'd have a bunny with kind of a medium fuzzy fur texture. But the most common one is this Snapdragon example, and I've got pictures to go along with it. So if we cross together a red one and a white one, you're going to get all pink ones, right? So if you are a heterozygote, you have a completely different phenotype than the homozygotes of either color. Right here, we have another one. So if this is big R, and this is little r, big R. If you have a mixture, you're going to be somewhere in between. And if you have both alleles, you're going to have the same color. So that's incomplete dominance. Nothing too crazy. It is really, really easy to get incomplete and codominance mixed up. So if you ever think of a mnemonic device, hit me up. I am a big fan of little stuff like that to help you remember. Because codominance is when the two alleles each affect the phenotype in separate, distinguishable ways. So if something was incomplete dominance, right, you have the mixture of the two. But codominance is where like some cells have one phenotype and some cells have the other. So codominance there shown together, essentially, that's where we get stuff like spots. So I don't have a written out example here, but if if a black chicken and a white chicken are crossed together, the offspring would be speckled, so black with white spots. So incomplete is like a mesh between the two. Co is spots, essentially. At least when we're looking at like fur phenotypes. Yeah. So if we've got one with both black alleles, one with both white alleles, the heterozygote will be speckled. Same thing with this weird fish example here. If you've got a red fish and a blue fish and color is inherited via codominance, then you're going to get spots. Cool? Incomplete codominance? Okay. Yes. So, the next two slides get kind of wordy. So, this is a real big practice in shorthand. So, for any characteristic that we observe, right? Dominant and recessive relationships really depend on the level that we observe them. So, like a lot of things in life, our perspective is what affects the label. So, the example that I'm going to give here is Tay-Sachs disease. So, just as an example or some background information, Tay-Sachs disease causes an inability to metabolize certain lipids because an enzyme doesn't work properly. As the lipids accumulate because they can't be broken down, a child with Tay-Sachs disease can have seizures, blindness, and then degradation of motor and mental performance, and often doesn't live very long. So because their body doesn't produce the proper enzyme to break down these lipids, these lipids kind of accumulate. So that's our background on Tay-Sachs disease. How it's inherited is interesting. So if only a child with 2 copies of the Tay-Sachs allele is said to have the disease. So they have to have both copies of the faulty gene in order to considered to have Tay-Sachs disease. So if we're looking at the level of the organism, right? It's going to be recessive, right? If you have both recessive alleles, you have the disease, but if you only have one, you don't. So just looking at whether or not someone has a disease or not, it's our Mendelian complete dominance. Yeah? Let me see that. Okay. If you look a little closer with the activity level of the enzyme, right? Our heterozygote has an intermediate level of activity. So if the enzyme works to full effect with both alleles, but half effect with only one allele, right? It doesn't cause Tay-Sachs disease because the small amount of function is still enough to not cause any outwardly symptoms. But because the activity level is somewhere in between, we've got an intermediate phenotype at the biochemical level. So if we're looking at enzyme activity, we would say that doesn't look like complete dominance, that looks like incomplete dominance because our phenotype is halfway in between. Right? So if we're looking at it as a biochemist, this would be more incomplete dominance. That makes sense? If you look even further, though, we can see that some cells produce an equal number of normal and dysfunctional enzyme molecules. So some cells produce our normal ones, and some cells produce our faulty ones. So when we're looking at a molecular level, that's more codominance because we have some good ones and some bad ones. It's not a half-working regular one. If we're looking at a molecular level, we've got codominance. So everything is subjective, essentially, and the level at which we're looking at phenotype can affect what our understanding of the effects of that genotype may be. So if somebody asks about the inheritance of Tay-Sachs disease, if they're talking about at an organ or at the level of the organism, we've got a simple Mendelian complete dominance. If we're looking at an enzymatic biochemical level, we're seeing incomplete dominance, but at the molecular level, we're getting codominance. So usually, whenever you are presented with a problem, you are given whatever phenotype you are looking for. So that kind of tells you what level you are observing things. But this is just kind of biology is a lot more complex than you think it is. Everything can be seen from a different perspective. Frequency is also something that we will talk about a lot as we move forward. So a lot of people assume that if a dominant allele that a dominant allele would be more common than a recessive allele. That if something is dominant, you're going to see it more in a population than if you weren't, because or then you would see the recessive allele, because if you've got the allele, you're gonna have the trait, right? But it's not always the case. For example, polydactyly, so being born with an extra finger or extra toe, is caused by a dominant allele. So if your parent had polydactyly and has 6 fingers on one hand, odds are you are going to, goodness gracious, if they have an extra finger on one hand, you've got a 50-50 shot of getting an extra finger on a hand. Because it's a dominant allele, you're going to have a 50-50 chance if they are a heterozygote. Because if they have the trait, you are going to have the trait. But in reality, only 1 out of 400 babies are born with an extra digit. So even though polydactyly is a dominant allele, the phenotype isn't necessarily common. And we'll talk a lot about allele frequencies when we hit farther down the line in genetics or tinkering on the lines with populations. So population genetics is going to be a lot of allele frequency stuff. And we'll get there. But just to kind of set you in place. So that's allele frequencies. So far, all of the traits we have talked about only have 2 alleles, right? There's a big B and a little b, a big Y and a little y, a big P and a little p. But in most genes, there's more than one allele that controls a phenotypic outcome. For example, ABO blood groups, right? This isn't this isn't like the positives and negatives. We'll get into that later. But whether or not you have A blood, whether or not you have B blood, whether or not you have AB blood, whether or not you have O blood, is determined by 3 different alleles. And I'll do some practice stuff on the next slide. So in a lot of genes, we have more than one allele, right? It's not just brown hair and blonde hair. It's not just brown eyes and blue eyes, right? There are more than one or more than 2 different phenotypic outcomes. We can have a lot of different things show up. We have a lot of different alleles for certain traits. And ABO blood groups is usually the most common example. The 3 alleles code for carbohydrates that are on the outside of the red blood cells, and that just kind of helps us identify. That's why whenever you get a blood transfusion, you have to have the right type of blood, because if you have A blood and you are given B blood, your body doesn't know how to identify B blood because it doesn't know how to identify those B carbohydrates. So we've got some examples here. The alleles for ABO blood group are written with I. The I stands for isoagglutinogen. It's just the name of the class of protein that are on there. So it's isoagglutinogen A, B. So there's 3 different alleles. You can have an IA, an IB, or a lowercase I. When you have the IA, you're going to get this A carbohydrate. If you have the IB, you're going to get this B carbohydrate. And if you have the lowercase I, you're just not going to have a carbohydrate. They show a codominance inheritance structure. So if you have IA, IB, right, you're going to have both of them. If you have IA or IA little I, you're going to have type A blood. If you have IB or IB little I, you're going to have B blood. The only time you are going to have O blood is if you have both little I's. Have you guys worked with blood typing question genetics before? Okay. It's a pretty common example. We'll probably run into it a lot. Here's another one. So, if you are blood type A, right, you're only going to have these little A antigens or carbohydrates, which means your body has antibodies for type B, which means it will attack type B blood if it enters your system. So if you have type A blood, you can be transfused type A blood or type O blood, because type O blood doesn't have anything on the top of it, which is why O blood is considered a universal donor. So if you have type O blood, specifically O negative blood, and you donate blood once, the Red Cross will send you emails every 2 days trying to get some of that, because it is the most helpful. If you have type AB blood, you can accept any of the blood types. So you are considered a universal acceptor, because you can have a type A antigens, you can have type B antigens, or you could have type O antigens. So if you are in an emergency situation or if someone is in an emergency situation and they need a blood transfusion, they are automatically going to get type O blood um, if there's not enough time to blood type them beforehand. Let's see. Does anybody have any questions here? Do you want to do a practice problem with it? All right. We will definitely interact with this in the future. Another type of complex inheritance is called pleiotropy. Pleiotropy is when one gene has multiple phenotypic effects. So if you have one allele, it's going to do many different things. Humans are complex organisms, and all of the information that makes a human is going to be stored in their DNA. So we're going to reuse DNA to do multiple things. Our example here is hereditary diseases, right? If you have, or if someone has a disease that they inherited from a family member, odds are it's going to have multiple symptoms. So cystic fibrosis and sickle cell disease are both affected by one allele, but they have a lot of different effects on a body. So one allele can have a lot of different phenotypic effects. In pea plants, that pleiotropy also exists. So the gene that determines flower color, which is a gene we've interacted with a lot with simple Mendelian genetics, also affects the color coding on the outer surface of the seed. So if you have one lighter color, you'll have a different color coding on the outside of the seed. So it is really just pure luck that Gregor Mendel didn't choose that coding as one of the traits that he studied, because we wouldn't have gotten the clear results that he did. Oh, you're going a lot faster than I thought we would. Everybody doing okay? Okay. So this extends to more genes a lot. So a lot of the times having one gene will affect another. So in dominance relationships, multiple alleles, and pleiotropy, they all have to do with the effects of alleles on a single gene, right? So the gene for flower color, the gene for sickle cell disease, they're all one allele on one gene. But a lot of the times we have two or more genes that involve developing a specific phenotype. Human height is one of those examples. There are a lot of different things in our genes that go together to like make us as tall or short as we are. So epistasis. Epistasis is the phenotypic expression of a gene at one locus that alters a gene at a second locus. So our example here is in Labrador retrievers, right? So we're going to look at black labs, chocolate labs, and the golden ones. So in labs, black is dominant to brown. We got simple Mendelian genetics. If you're big B, or big B, little b, it's going to be black. If it's little b, it's going to be brown. But there is a second gene that determines whether or not that black or brown pigment will even be deposited. So a dominant allele E results in the actual deposit of the pigment. But if the dog is homozygous recessive, so little e, then the second gene, or at the second gene, then the coat's going to be yellow, because regardless of if it has a big B or a little b, if it can't deposit the color, you're going to get a yellow lab. So that second gene affects what happens to the first gene. Does that make sense? I think I've got a little chart. Yeah. So even though this is a heterozygote, right? And this is a heterozygote, we're not getting our 9:3 to 3:1 ratio, because that's only with our Mendelian genetics. So if we've got a big E, big E, big B, little b, right, we're going to have a black lab, and the color is going to be deposited. Down here, we have big B, little e, little e. If we were just looking at the first two letters, would that dog be a black lab or a chocolate lab? Black lab, because we got our capital dominant alleles. But because we have our two recessive alleles, right, this one's going to be little e, or it's going to be yellow. Same thing down here. If you have a little b, little b, little e, little e, it would have been a brown dog, but without that color deposit, we've got our yellow lab. So it is very possible to cross a brown lab with a yellow lab and get black black labs from that cross. If that black allele was hiding in the yellow lab's genetics. Does that make sense? Okay. So it's possible to cross a chocolate lab with a yellow lab and get either yellow labs or black labs, but not chocolate labs. And that's just because we have one gene, the E gene, that affects whether or not the black or brown gene is actually going to be expressed. The next is polygenic inheritance. So a lot of genes, our examples are human skin color and height, are not of two discrete characteristics, right? It's not you're 6 foot or you're 5 foot. There's a big gradient of heights that you can be. And it's not just really, really dark skin and really, really light skin. There's a whole gradient of human skin colors in between. So the population, the characteristics vary within a population among a continuum. These are called quantitative characteristics. Usually, having some sort of gradient in a phenotype indicates polygenic inheritance. And polygenic inheritance is an additive effect of two or more genes on a single phenotype. So, like the converse of pleiotropy. So, in pleiotropy, right, we had one gene affecting multiple characteristics. In polygenic inheritance, we have multiple genes affecting one characteristic. Does that make sense? So, for example, I am a rather short human being sitting at like 5'3", 5'4". My sister, I think, is an inch taller than me, but my brother, who's 14, is already a good 6 inches taller than me. So even though we've got the same genetic information to pull from, we've got already, just with 3 people, a decent spread. I am also a very, very pale human being, but my sister can go out in the sun for an hour and a half and get really, really tan, no problem, whereas I will peel like a snake. So, different genes affecting different alleles, or different genes affecting the same alleles. So, we have different genes coming together to give us different heights, different genes coming together to give us different skin tones. These genes also exist in organisms that aren't humans. Humans are just the easiest example because we see the most of them. So height and skin color are usually our two big examples for polygenic inheritance, but there's a lot more. Eye color is actually polygenic inheritance. There's a lot of different alleles that go into eye color, or a lot of like different genes, that go into whatever eye color is being expressed. So a human example, like I said, is height. In 2014, they did a study with over 250,000 people, and found 700 genetic variations associated with over 180 genes that affect height. So there are over 180 different genes. Ooh, that's not a word. 180 different genes that affect height. So this is variations in genes involved in pathways that affect the growth of our skeletons. So there are different pathways that affect how our bones grow, and those pathways are controlled by genes. But there are other genes in this 180 that are associated with stuff that's not super obviously related to growth, but they do, in the end, affect height, which is really cool. Ooh, this one's fun. Environmental effect on phenotype. So this is kind of where nature versus nurture comes into play, right? So sometimes the phenotype or the physical qualities of an organism depends on the environment as well as the genotype. For example, if you had two identical trees, right, and one grew up in a very windy, very sunny, very rainy scenario, and another one kind of grew up in a shaded, less windy scenario, they're gonna have different leaf sizes, different levels of greenness, um, different heights depending on the exposure to wind and sun. So just because you have a tree with the genetic capability of being gigantic, it doesn't necessarily mean that it's going to have the resources to become gigantic. Also in humans, so your nutritional value in your youth will often affect how tall you are. Um, Exercise can alter the shape of your muscles, your build. Sun exposure can affect your skin tone. Um, Experience and practice improves performance in intelligence tests. Literally, scientific genetic proof of how growth mindsets are more beneficial, because if you practice more, you'll learn more, and you aren't born smart, you aren't born dumb. Everybody's got a starting point and can work up to wherever they need to be. I got a couple examples that I think are really fun. So in these hydrangea flowers, the colors range from blue to pink, and that's not genetic as much as it is a reaction to the acidity of the soil. So if you have hydrangeas that are planted in very basic soil, you're going to get pink flowers. If you have hydrangeas planted in acidic soil, you're going to get blue flowers. But you're only going to get the blue flowers if you have free aluminum in the soil. So even if it's acidic, if there's no aluminum available, you're still going to get pink, which I think is super cool. This one's my favorite. Himalayan rabbit coat color is determined by temperature. So if you go out in the cold, right, the first thing that gets cold is your fingers, your ears, your toes, your nose, right? Because that's the outer end of our limbs. If you look at a Himalayan rabbit, right, it's the toes, the nose, and the ears that's the darker color. Because if something is a darker color, right, it's going to absorb more heat. If you're out and about with friends in the middle of the summer, you got a big group going. One friend has a black car, one friend has a white car. You're going to want to get in the white car to go from place to place, because you know that black car is super, super hot on the inside, because black absorbs more heat. So if a rabbit is around or if a rabbit grows its fur during the winter time, right, wherever it is cold, it's going to exhibit a black phenotype. But if a rabbit is born during the summertime, in really, really warm weather, it's going to look all white. There's an ongoing thing, I think, with all biology teachers, that if you had a class pet that was a Himalayan rabbit, you could shave off the hair on its back, strap an ice pack to it, and the hair that regrows in that area is going to be black because it was exposed to cold temperatures. Because the environment affects phenotype. Do we have any questions here? So generally, phenotypic range is broadest for polygenic characteristics, and the environment can also affect the range at which things is. Skin color is a really big indicator here. So someone can have the genes to be darker skinned and the genes to be lighter skinned, but the exposure to the sun can also affect that. And these characteristics that can be affected by genetics or the environment are said to be multifactorial. So, meaning there are multiple factors that influence phenotype. We got through that a lot faster than I thought I would. If you have any questions with any of this, please let me know. The next slide does say practice questions on Canvas, but there are not yet, because that's what we are going to do tomorrow. But I'll try to think of something to spice it up a little bit so we're not just sitting. Yeah, if you have any questions about this, please let me know. Genetics is super cool, and there's a lot of stuff that goes into it. Slideshows more than... Gotcha. All right, cool. Yeah, my only issues with PowerPoints is my pacing. I'm still trying to figure out how fast I should go. But the more we do it, the better I'll get. Beautiful. Sorry. Cool. Here, I'll bring this back. Sorry.